This image is so incredibly important because in this image, we see ovulation all the way to implantation. So day 0, essentially, once fertilization occurs, all the way to day 10, when implantation in the uterus has occurred. As you can see here, day 0 is fertilization. The unfertilized oocyte has made its way a few centimeters through the fallopian tube. Fertilization has occurred. Then the zygote makes its way down the fallopian tube. But what's interesting, as this movement is occurring down the fallopian tube, all sorts of processes are occurring with that zygote. For example, we have the first cleavage division, where that fertilized egg, the zygote, goes through its first division, and we've divided the DNA. So now we have the 2-cell stage, which is each one of the cells is called a blastomere. Then those 2 cells divide. This is about day 2. We're at the 4-cell stage. Then we get to day 3, where we have this early morula. Looks a little bit like a soccer ball, except it's solid on the inside. Then day 4, we have the advanced morula. Then day 6, we have the blastocyst. But as all of these divisions are occurring, and these changes are occurring, because when you see day 6, you see clearly some cells are doing things that are different from the other cells. So we have different jobs occurring even while we're having this division occurring. All right, so let's talk about this in a little more detail. Blastomeres are the identical cells that are formed from mitotic cell division. The morula is the structure that is formed after multiple cell divisions. And here is a picture of a morula. Remember I said it looks like a soccer ball. However, it is different because it is solid on the inside. So in the text here, I tell you that it resembles a mulberry, and not very many people know what a mulberry looks like. So let's say it looks like a raspberry. You know how raspberry is hollow in the very, very center? So it has to be a solid raspberry. It doesn't have that hollow spot. Once you have all of the blastomeres formed together, they actually form what is called a blastocyst. So this is the stage that occurs after a morula. So this is a blastocyst, and it has distinct areas. It has the inner portion that is called a blastocele. It has the inner cell mass here that is formed at one end. And then you have the outer cells that make up the trophoblast. The inner cell mass, as I circle here, is found at one end of the blastocyst. It's exposed on one side to the space, which is the blastocele. It's insulated from the outside environment by the trophoblast that you can see me marking right here. And later, this inner cell mass will form the embryo. Implantation occurs about 7 days after fertilization. This image is a picture of what happens when implantation occurs. So the blastocyst that I pointed out earlier, located right here, makes its way on or about day 6 into the uterus from the fallopian tubes. As you can see, the endometrium, the functional zone, is presented here. It is highly vascularized, very thick. As I've referred to it before, it's a nice, soft, cushy place for implantation to occur. So this blastocyst is going to eventually, on or around day 9, is going to implant into the uterine wall. When this occurs, in order for implantation to occur, those trophoblastic cells that are on the outside are going to go through rapid division, and they create several layers. So not only do we have the general term for trophoblasts, but we have what are called cellular trophoblasts. So if you'll notice, the inner cell mass has polarity. Let me start that again. The blastocyst has polarity because we have an inner cell mass on one side and it is absent on the other. The inner cell mass rotates itself so that it is facing the functional zone of the uterus. The cellular trophoblastic cells are these cells right here. They're closest to the inner cell mass, and they are the cells that initially touch the endometrium. All of these trophoblastic cells go through this rapid division, and another layer that's formed is called the syncytial trophoblast. So these are the syncytial trophoblastic cells. They are the outer layer, and they will secrete hyaluronidase. And if you remember, we've seen hyaluronidase before. That's one of the enzymes in the acrosomal cap of the sperm. So this blastocyst actually will secrete hyaluronidase as well. And it has somewhat of the same job. It will erode the path through the uterine epithelium, right? And that's what you're seeing right here. It's this erosion of this path through the functional zone of the uterus. So once we have been fully implanted in the uterus, then the differentiation continues of what was originally the blastocyst. And the inner cell mass, which was this structure right here, will begin as two layers. This superficial layer that you can see right here, and it faces the amniotic cavity, which is right here. And then there's the deep layer that's exposed to this space. We have migration of the cells around the amniotic cavity, and that's the first step in formation of the amnion. And the migration of these cells is the first step in making this yolk sac. So we still are we are still making our way in the functional zone of the endothelium or endometrium of the uterus. We still have the syncytial trophoblast, we still have the cellular trophoblast, but now we have other layers. One of the things that you'll notice from the syncytial trophoblast is we have these villi, plural, that are forming, right? These little finger-like projections that are forming. So we have this inner cell mass. Saw the superficial layer, you saw the deeper layer. And then as we're continuing on day 12, something called gastrulation will occur. And this is the formation of a third layer of the structure. So that third layer is called the mesoderm. So you have these superficial cells that form this third layer. They're randomly dispersed through here. So first layer, second layer, this inside is the third layer. And these migrating cells will move toward what is known as the central line, right? And also known as the primitive streak. That's called gastrulation. So now we're at about week 2, and those mesodermal cells are going to migrate to the inner surface, and they're going to form what is called the chorion, which is composed of the cellular trophoblast and the mesoderm. Right. So this embryonic disc is formed. It's a 3-layered sheet. It's produced by gastrulation, and it forms the body of the embryo. The rest of the blastocyst will help form the extra embryonic membranes, and we'll talk about those in just a minute. So this embryonic disc, it's bulging in to the amnion. There is a head fold that you can see right here. We have something that's called the allantois. It's an extension, and that extends toward the trophoblastic cells, which are around here. All right. So those extra embryonic membranes, there are 4, and that includes the yolk sac, the amnion, the allantois, and the chorion. All right, so first, let's look at the yolk sac. It's first a layer of cells that forms around the outer edges of the blastocele, and this is where blood formation occurs. The next structure that we want to look at is the amnion. So that's the combination of the mesoderm and the ectoderm, right? Combination of these two, and the cells spread over the inner surface of the amniotic cavity. So this is your amniotic cavity, and these cells form this amnion, and it enlarges all the way through development. Amniotic fluid, right? So here's your amnion. It has amniotic fluid in here. It's found in this cavity, and it helps the embryo and fetus by cushioning it. All right, the allantois is this branch-like structure right here. It's actually a sac of endoderm and mesoderm cells. They give rise later to the urinary bladder of the embryo. The chorion is all of this right here, right? Everything that you see here in pink. The chorion is mesoderm cells and trophoblast cells, and there are blood vessels that develop within the mesoderm, and so this becomes a rapid transit system for nutrients that are linking the trophoblast, right? Remember, this is the trophoblast with the embryo, right? So this is the developing embryo. So this chorion is the first step in the development of a functional placenta. Earlier, I pointed out to you the villi, and I'm showing them to you here. These are finger-like projections. They are in contact with the maternal tissues. So this contact with the maternal tissues allows for this network to form, and it carries maternal blood, right? These are the chorion villi. Once we get to week 4, as you can see, we have an embryo has the head fold right here, and it has the tail right here. And there are constrictions between the embryo and the trophoblast, and this body stalk is going to narrow. Yolk stalk and the body stalk are going to narrow. When we get to week 5, the embryo bulges out into the uterine cavity, because remember, this is the uterus. We have the body stalk and the yolk stalk have fused, and they become the umbilical stalk, which will become the umbilicus, or the umbilical cord. Skipping along to week 10, the amnion has clearly expanded. You can see the amnion right here. It fills the uterine cavity. The fetus is connected to the placenta by the umbilical cord that you can see right here. And it has a portion of the allantois, the blood vessels, and the remnants of the yolk sac that are located here. So embryogenesis is the term used to describe the development of the embryo. And this is when the embryo starts to separate from that embryonic disc that I showed you earlier. You can see that the internal organs start to form, and folding and different growths of different areas of the disc indicate that we will have a head fold and a tail fold. When we are at week 4, even though it was a very simple cartoon earlier, here's the forebrain, here's the eye, here's the ear. Looks like it's in an odd place, but it'll eventually come together. These are the pharyngeal arches, or the gill slits. this is the two components of the heart. Here's the tail. Yes, everybody has a tail at some time in their life. There's a leg bud, and there's an arm bud. So you can see by week 4 that many of the structure, you can actually recognize many of the structures. So I put this picture in here just so that you can see embryologically when we are in development. A human looks very similar to other mammals such as rabbits, cows, and pigs, to birds such as chickens, to tortoises, and to salamanders, and to fish. So we all have gill slits. We all have brain development. We all have eye development. We actually have spinal cord development for all. So we all look quite similar. So when we think about development, and I think about the questions that I've gotten over the years, let's just think about twins, and let me see if I can dispel some misconceptions. All twins come from one sperm fertilizing one egg/oocyte, all of them. Now, what happens after that fertilization event occurs could lead to some different types of twins. We have monozygotic or identical twins. This means you have a single fertilized egg, a single zygote. It splits into 2 developing zygotes at a very early stage. These individuals are identical, therefore they are going to be the same sex. If the split doesn't occur as cleanly as one would like, then perhaps these individuals will end up being conjoined at some space. Dizygotic or polyovular or fraternal twins is still one sperm fertilizing one egg, but the mom ovulated 2 or more eggs, and so there are 2 or more. We had fraternal triplets, let's say, that would be 3 eggs were ovulated, or 3 oocytes were ovulated, and 3 separate fertilization events occurred. That's a very, very rare. These individuals can be different sexes. They're no closer genetically than any other siblings. My brother's 3 years older than me. If we had been born at the same time, we would be fraternal twins. We still would not be identical, right? Interestingly, the incidence of this occurring increases with the age of the mom. So we know that a human female is set up to have a single infant. But as we age, some of our systems begin to fail, and so a woman will ovulate more eggs as she ages because we just don't have the system in place to stop that. Sometimes fertilization events don't get it right. If there's 2 little sperm, as we've talked about before, it could lead to infertility. And remember what I said to you early on, how many sperm does it take to fertilize an oocyte? Many of them. Sometimes there's too much sperm where you can get dispermy or triploidy. That's a polyspermy event. And remember what I said when we talked about polyspermy earlier. And it is a rare, rare occurrence that an individual that has arisen from polyspermy will become full term. Most of the time it leads to spontaneous abortion, otherwise known as a miscarriage in most cases. Another way fertilization can go awry is in a bad location. If that fertilized egg makes its way out, either makes its way out of the fallopian tube or does not implant in the uterus, maybe it implants in the fallopian tube. That is called an ectopic pregnancy. Anytime a fertilized egg implants anywhere but within the uterus, it is called an ectopic pregnancy. If a woman has endometriosis, where the endometrium is overgrown, then she could have endometrium, and her endometrium could actually go up into her fallopian tubes. If that happens, then there's the fertilized egg doesn't know where to implant. It doesn't know the difference between the fact that that is the fallopian tube versus the uterus, because it has everything that it needs to implant. Sometimes the endometrium can grow outside of the uterus, especially if a woman has a fistula in her uterus, or if the endometrium has grown all the way through the fallopian tubes. That can lead to an ectopic pregnancy. Another way that fertilization can go amiss is if there's bad timing. Sperm only lasts for 48 hours within the female genital tract. Remember, it is called the hostile environment of the vagina. And an ovulated oocyte can't be fertilized after 24 hours. So ovulation and the presentation of sperm have to occur within that 24-hour time period.